this whole entire video is going to be on honey. It has a lot of different effects. So starting with calreticulin on cancer cells, that's the CRT. So that's actually the eat me signal. When expressed on the surface of cancer cells, it flags them for phagocytosis or engulfing by immune cells. Now the problem in tumors is that many cancers they downregulate the CRT so that it's sequestered on the inside of the cancer cell. That means that the immune cell can't see the cancer cell because the CRT has to be on the outside. And the way the macrophage does the phagocytosis is with the CD91 receptor. The CD91 recognizes the CRT. And now honey has some potential effects with its polyphenols and its flavonoids, and it can actually enhance macrophage activity potentially by increasing the CD91. So basically, that means that it's that honey helps the macrophage phagocytosis on the cancer cell by increasing the CD91. Honey also it may also help macrophages target CRT by reducing the excessive inflammation while still promoting the effective immune surveillance. Now, you might want to be careful with the honey, because some honeys have been found to have glyphosate. Yeah, glyphosate can be a bad thing. It's basically a pesticide that farmers use. How the glyphosate got in, I don't really know. But I think some brands of honey don't have it. So you're going to have to do some research on the brands that don't have the honey brands that don't have a glyphosate. Honey also has effects on the CD47 and a SIRP-alpha axis. CD47 is the don't eat me signal that cancer cells express. So basically, what that does is that there's a SIRP-alpha or SIRP-alpha that's expressed on macrophages. What the CD47 on the cancer cell does is that it binds onto the SIRP-alpha on the macrophage, inhibiting phagocytosis. But honey can actually enhance phagocytosis by actually counteracting the CD47, modulating a reactive oxygen species. Honey also encourages M1 anti-tumor as compared to M2, which is pro-tumor. So, tumors can have the ability to skew towards an M2 phenotype, which is actually a bad thing. But the goal is to switch it over to an M1 phenotype, because that's where the tumors can't survive under an M1 phenotype. And M2 does have its benefits, but in a cancer, but when it comes to a cancer context, M2 is a bad thing. So the goal is to go to M1, and honey can help with that. There are some other foods that can also help to skew towards M1, but some other foods, even health foods, can actually skew towards M2. So that's where you have to be careful, which supports cancer to survive, which I'll have to cover in a different video. It's beyond the scope of this video. So basically, honey doesn't block the CD47 SIRP-alpha binding, but honey can enhance macrophage activity and modulate cytokines. Honey has quercetin. Quercetin can deplete zinc so that it's not excessive. If quercetin is excessive, if zinc is excessive, then that's going to reduce copper. And then less copper means less iron, and then there's going to be less iron for the red blood cells, and that means that the red blood cells can't deliver oxygen, and then that means that there's going to be a hypoxia, which is a low oxygen, and that's how cancer cells survive, or they grow from hypoxia. Hypoxia also supports the mechanisms that cancer cells use to recruit tregs. So yeah, quercetin in honey can help reduce that by reducing excess zinc. Excessive zinc also closes chromatin, And if chromatin is closed, that's where the DNA is, that the gene transcription factors that are needed for the immune system to function will also be disabled. Honey can help keep chromatin opened for the immune system to work by reducing excess zinc that would otherwise close the chromatin, using quercetin to reduce the zinc. Honey is also good for the goblet cells because the goblet cells secrete mucin, and mucin is needed to keep the mucosal layer of your stomach, of your gut, closed. That's important to prevent lipopolysaccharides from leaking. They leak into the bloodstream. Lipopolysaccharides can also, in some cases, lead to sepsis. Lipopolysaccharides are also inflammatory, and that's how cancer survives under inflammatory conditions. Lipopolysaccharides also bind to toll-like receptor 4 on macrophages, which can lead to more inflammation. So, yeah, honey can help goblet cells in the gut so that the goblet cells can secrete mucin. Goblet cells are very sensitive to oxidative and inflammatory injury. Honey compounds decrease NFKB. So this means that inflammation is reduced. Honey and its compounds reduce cytokines such as IL-1 beta and TNF alpha. These normally deplete goblet cells or cause goblet cells to do apoptosis. So if the goblet cells aren't there, then that means that no mucin is going to be secreted, which means that you are going to have a weak mucosal layer. Lipopolysaccharides or gram-negative bacteria are going to be leaked and cause problems. They're going to cause more inflammation. So honey maintains healthy goblet cell density and prevents a chronic mucin depletion in the gut. Now, honey also regulates mucin gene expression. Honey also stimulates epithelial reconstruction by increasing AMPK and ERK repair signals. Honey also enhances tight junction proteins called occludin and claudin. Now, these are important. These have to be kept tight. These are proteins, those proteins have to be enhanced so that the gut mucosal layer stays tight. Because without them, without those proteins, occludin and claudin, then the gut is going to be weak, and then it's going to release gram-negative bacteria, and it's going to cause its problems and inflammation. Now, honey, especially Manuka, can soothe the airway epithelium, and which is a slight increase in mucin hydration. It also increases short-chain fatty acids, such as butyrate, which has a lot of health effects that could actually go beyond the scope of this video. So I'll have to make a different video for that one. So subscribe if you have any insights, any knowledge, any mechanisms, or any foods that you know about that can help cancer or even support the gut. Leave those down in the comment, and I'll see you in the next video.